talk about bacteria making nanomaterials as opposed to a chemist. And I'm going to try to convince you that bacteria can be as good as chemists and physicists to make such materials. Okay, so my group's been doing metal microbe interactions for some time. Initially, we were mostly interested in looking at metals as alternative to organic antimicrobials, and we're seeing uses in wound infection control, antibacterial, antibiofilm coatings. We've done a lot in basic environmental pollution, bioremediation of metal and organic polluted sites, as well as recently we've gotten into corrosion control and anti-biofouling. Today I will concentrate on the nanomaterial aspect of the bacteria bioconverting the metal ions into precipitates, and then some characterization of their antimicrobial, electrical, and fluorescent properties. So a little bit of background on metal microbe interactions. We had the opportunity to write a couple of reviews in this area, looking at how bacteria can be tolerant and resistant to metals. And I just kind of like this cover photo that nature gave us, where resistance is not futile. You get a little bit of growth there, but microbes, of course, have co-evolved with the during the Earth's geological changes, seeing high loads of very toxic metals at different times, and therefore have involved resistance. So there's a simpler cartoon to kind of show you how microbes deal with metals or how they're toxic. So if they're toxic, you can have the metal destroying, targeting a specific protein, destroying it. You get a variety of reactive radical species produced that can go off and do damaging chemistry. You can have cytotoxic effects with regards to reactions to the DNA. You can get holes poked into the membranes of the bacteria, arresting metabolism. Bacteria have generated resistance by simply preventing the metal from getting in. If they do get in, you have a possibility of it being spit out, an efflux system. You can repair damage, you can repair the proteins, and produce chemicals that will chelate the metal and protect. So basic chemistry behind metal microbe interaction, the biochemistry, through our analysis over 20 years, we basically see it following hard, soft, acid, base theory, where our metals from the periodic table can be grouped into soft or hard acids. Here's all your functional groups that are in biochemistry. So if we were looking at silver, for example, we'd expect a interaction with thiols. And what you tend to see is increased toxicity as you go to this side. So these metals are generally more toxic than on this side. So your atomic radius, oxidation state, electronegativity, polarizability, all these parameters on how they trend with regards to the toxicity. Something else we consider is the Irving-Williams series of metal ligand binding, where we have to recognize that copper has a more flexible binding coordination, and it can replace other metals, and that helps copper, which is actually borderline, actually is fairly toxic as well. How we got into this area, again, looking at bioremediation, we were working with microbial biofilms from mine tailings, and what you're seeing here is a scanning electron microscope, and this is bacteria in the background in both of these pictures here. In this picture, these are all bacteria. We hit these bacteria with high loads of metal ions, and we see the bacteria catalyzing the crystals of the various metals to generate basically nuggets of the metals. So here we're seeing examples for copper, silver, and strontium, and these are on the micron sizes, these crystals, but this led us to the idea that we can convert biomining to a combination of into remediation towards precious metal recovery. an issue that's of interest in Europe for sure. So background for nanomaterial synthesis, we've already heard some great talks this morning where you see the chemists and the physicists using harsh chemicals, extremes in pH, extremes in pressure, and extremes in temperature. Can we go to a more eco-friendly approach to generate our nanomaterials? Organic waste products such as agricultural waste have been explored for this. And in fact, just the reducing sugars from plant material can be used to synthesize silver and gold nanoparticles. And that has been explored for even in the early 1990s and then through the 2000s. This is a process, though, is very difficult to control from reproducibility issues. So we've seen an increased interest in using bacteria for nanoparticle production. So that's where I came in. And it all kind of started when I did a sabbatical in Professor Zanoni's lab in Bologna, Italy, and we were exploring some metal microbe interaction. And this has led to wonderful collaborations with the groups in Verona as well, with Giovanna Bellini and Silvia Lampis. And they send me PhD students every summer. Apparently Italians don't want to come to the north of Canada in the cold, in the winter time, but they spend 6 to 12 months in my lab working, and I get to go to Italy. So I'm going to talk about mostly Elena and Alessandro's work, who actually joined my group on a more permanent basis. Where does this all begin? In the early 1990s, the oxyanion form of tellurium tellurite was used in selective media for identifying enteric pathogenic E. coli. And you see here bacteria, black dots here. These are colonies on a media plate, black culture. And it was identified that the pathogenic E. coli actually had a resistant determinant for tellurite. And we see here, we have precipitates of the tellurium within the cell kind of crystallizing along inside the membrane, which leads to the black coloration. So in clinical labs, they said, beware the black E. coli. If you had black colonies, you had pathogenic enterics. Tellurite is actually very toxic to bacteria, more than mercury, arsenic, cadmium, and stuff, and other elements. The second time I observed this was, again, when I was working with Professor Zanoni. We were trying to work out the electron transfer chain in a photosynthetic organism, Rhodococcus capsulatus. And we were using tellurite to steal electrons away from that electron transport chain so we can poison it in different places. And again, we saw these crystalline precipitates inside the bacteria. So this was 2007. Moving forward, here we are 2014 when we're trying to do this on purpose, where we used a quinone analog that had a redox poise close to that of the tellurite reduction. And our idea was to steal the electrons away from the electron transport chain to generate our nanorods or nanocrystals that we're seeing here of tellurium. And this table here is just to show you that this was the first time we realized that by playing around with the metabolism of the bacteria, we could get different yields and slightly different structures of our nanomaterial. So this process then was to make our nanomaterial. We did it with selenium as well. We got selenium nanoparticles and then the nanocrystallines. And we're using our photosynthetic bacteria, using light energy to move electrons that we steal with the Lawson redox electron carrier for that reaction to make our material. So matching physiology with redox poise. This led us to explore with the University of Verona. They were interested in bioremediation of metal polluted sites. And we isolated a number of bacteria from a variety of selenium and mine polluted environments. And we also landed up using this bacteria strain BCP1, which we were using a lot for bioremediation. And my group identified that it is incredibly metal resistant. So the idea is if it's metal resistant, we can use a higher metal load, allowing us to perhaps make different nanomaterials and higher yields. So here we have just an example. We'd have a small culture here. We'd add our tellurite. The process of the metabolism would generate the selenium nanoparticles in this case. And it turns, depending on the physiology organism, you might see a coloration anywhere from a light yellowy orange to a very dark red, even to a purplish color. Here's just some of our earlier work where we're identifying with Stenotrophomonas, a extracellular nanomaterial being produced from selenium. Here you have an intracellular tellurite accumulation. Moving on, we started to play and figure out how to do things a little better. And this is a dynamic light scattering, giving a size distribution for the incubation time. And we made chemically synthesized nanomaterial, and we could say that we had relatively the same kind of distribution, and the biogenic ones are slightly larger in size. We have a zeta potential here, which is the charge density of the material, which gives us an idea of stability. And we found this organism could make it just as well, just as stable particles as we could as chemists. Here's a couple of other organisms. When we looked at the other organisms making selenium nanoparticles, here we see with time, we have the selenium nanoparticles getting larger and larger and larger, whereas for the tellurium nanoparticles with this organism, they stayed about the same size. We were just able to get higher and higher yields. So different organisms giving rise to different outcomes. Here we have a transmission electron microscope with the Stenotrophomonas getting control of the production. Before we saw very large conglomerates. Now we're seeing smaller and smaller or smaller nanoparticles. We have still a good, we have very good size distribution and a good stability size. So this is just scanning electron microscopy versus transmission to look at identify the material that's being produced extracellularly. Here with this one, we're trying to play with the size of the material. Usually for a chemist, we can play with size by the initial salt of the metal that we're adding. Here we see that as we increase the metal load, we actually got smaller particles. So we were able to control the size by simply adding with the concentration, with all the other parameters the same. And all of these had about the same zeta potential stability. So these again take some very unexpected differences compared to that of how we do it as a chemist. If we look at the chemical procedures of bottom up, top down, and template based, we can generate a lot, as chemists, you can generate a lot of interesting nanomaterials. There's just some electron micrographs of these materials. We wanted to know, can we trick our bacteria to produce the different types of nanomaterials, and can we produce them at the same quality as a chemist could? So we chose this organism, again, PCB1. It's a pretty robust organism. And it also, when it's stressed, it produces surfactants, natural surfactants, which allows us to trick to either generate nanoparticles or nanorods as the end structures, which allows the difference between three-dimensional growth and one-dimensional growth, because you'd have the rod covered by the nanosurfactants, so the atoms are only being added to the one end. So how do we control this? We have to think about the microbiology. We have our organism species type, our energy and carbon sources for that organism. How do we extract? How can we manipulate that? Separation of the cells from the particles, the organic material from the particles. We have to think of all the somewhat the biochemistry and chemistry, what solvents. Still, we're playing with pH, ionic strength, redox potential, various things. One thing about nanoparticles for stability to control size, if I'm synthesizing it, the chemistry or using a laser, I'd want to cap that material with some sort of compound that would then stabilize it so it wouldn't aggregate any further and help stabilize it. Well, the bacteria are producing the capping materials from their own biochemistry. So they're going to be capping proteins, lipids, fatty acids, nucleic acids, carbohydrates, metabolites, everything that would be in the metabolism of what's going on. To date, we've identified a few specific proteins that make sense that these might be the actual enzymes catalyzing the reduction process towards the material, and then those proteins become the capping layer for the stability. So here we'll show you some of the results we were able to get, and some first tries of making nanorods with this organism. We're starting to get quite nice long nanorods. They're being made inside the bacteria. We play with concentration and time, and we use stationary phase resting cells that are producing a lot of the biosurfactant. When they're not, when they're early in their growth, they don't produce much biosurfactant, and we're getting nanoparticles kind of hidden around in this. But we can, it follows actually fairly nice kinetics relationships to length for the concentration and the time of incubation, so that we now know that we can kind of predict if we're looking for we want 700 nanometer rods, we can choose the conditions quite nicely. It's a very nice relationship here. The quality of the material, here we're doing selected area electron diffraction. We extracted and purified some of the nanorods. You can see a really nice diffraction pattern here. Zoom in, and the unit cell lattice is extremely close to what is in the chemistry one. So we have really nice, these bacteria are really great chemists, making nice long nanorods that are of really high quality. We're trying to do the same thing to generate selenium nanorods. We're having a little bit of trouble separating the nanorods from the particles, but we're getting closer here. Let's take a look at some of the useful properties. We can think of these towards electronics, photoluminescence, chemotherapies, environmental pollution cleanup. So I'm just going to show you a few quick examples. I just have a few minutes left, I see. Here we just did a resistance test of adding the nanorods to a wafer and measuring the resistance. And you can see as we add the rods, we get decreased resistance. So we have very good conductivity. One thing that we were very surprised of in the literature, selenium nanoparticles are not considered to be very fluorescent. Here is a chemically synthesized selenium nanoparticle. Very, very little. This is just a bit of a Raman scattering peak, but hardly any fluorescence. You've got to really zoom in to see it. The biogenic ones are very nicely fluorescent. So they have a plasmin structure, plasmins that are not in the normal ones. So that organic coating from the bacterial biochemistry has provided that. So here's just, we've been playing around with different organisms, so two different strains, growing the organisms in different way. We get different fluorescent properties. We can get different fluorescent particles, and our excited state lifetimes are different, even when we have the same strain growing in different conditions. So these LB and NB, they're different media types. So what kind of carbon energy sources the bacteria have, it's sort of the differences of the nutrient broth would be like you're having a healthy fruits plate for breakfast, and the LB you're having bacon and eggs. So you're going to have a slightly different energy state after that, whether or not you had coffee as well. So it's the difference to the bacteria's energy. We've explored these particles for their antimicrobial activity. The lines up here are different controls. These are all chemically synthesized. These two are biogenic. This is the concentration of our particles. This is colony forming units. So the curve crashing down here is the killing of the bacteria. So here's Pseudomonas aeruginosa, Staph aureus, two pathogenic strains, and we were able to kill off both planktonic and the pathogenic biofilm version with the selenium nanoparticles produced by the bacteria, not those that are chemically synthesized. So in summary then, we're taking an approach of removing the toxin. So we have selenium polluted sites in North America and Europe, tellurium polluted sites. We can take these bacteria, do bioremediation, take these toxic oxyanions out of the aquifer and have them bioconverted into interesting value-added nanomaterials that can be used for fluorescence, antimicrobial coatings, and things like that. So we have control of what kind of nanomaterial the bacteria can make. We're optimizing our conditions and starting to learn the actual biochemical mechanism behind it so that we can control it even more. Here's my team of Alessandro and Elena, which did most of the work here. Greta, she did some of the biochemical mechanism stuff I didn't talk about. And Eva here is actually making arsenic nanomaterials. Nikhil has just started, and he's playing with the BCB1 strain, making heteroatom nanomaterials that I haven't had time to talk about as well. And I have a team that's actually just continuing looking at mineral microbe interactions. So thank you very much.